Thank you everyone for attending the Science Matters seminar series today. We are pleased to welcome Dr. Daniel Doherty, Professor of Pediatrics and Head of the Pediatric Division of Developmental Medicine at the University of Washington. Dr. Doherty received his PhD and MD degrees from the University of California at San Francisco. He then joined the University of Washington, where he completed his residency and fellowship, later establishing his research lab in 2005. His research focuses on hindbrain malformations to better understand human brain development and disorders such as intellectual disability and autism. Using a range of genetic approaches, his lab identifies disease-causing genes and develops in vitro and in vivo models to study their molecular mechanisms. His team has discovered over 15 genetic causes of Joubert syndrome, helping to establish ciliopathies as a highly heterogeneous group of disorders. Today, Dr. Doherty will provide an overview of conditions linked to hindbrain malformations and take a deeper look at the genetics and mechanisms underlying Joubert syndrome. Thank you, Dan, for sharing your work with us.
All right, thanks for the kind introduction, and thanks to all of you for taking the time to come to the talk. I'm happy to field questions as we go along, although I might defer some to the end for the sake of time. So my title is somewhat intentionally cryptic for those who aren't familiar with human intracranial anatomy, but I'll tell you what the tentorium is in a minute. But first, I want to pause for a bit of context because it's front to mind for me every day. And that is that science, medicine, and education are under threat in the US and abroad, and we really all need to stay informed and advocate and educate pretty much everybody who we come in contact with because, in my opinion, our country is headed down a pretty dark road right now, and if we don't stay awake, we're more likely to end up where we don't want to be. I also like to call out that we do share this region with the descendants of Native American tribes who were here long before, or whose ancestors were here long before us. And finally, I think thanks or tangible actions speak louder than thanks. And so speaking up, voting, contacting your elected representatives, running for office if you've got the time, and supporting Native American businesses and organizations like Real Rent Duwamish are things that I would love people to consider. All right, so now to the science. The goals of my lab for the past 20 years have been to improve the lives of people with hindbrain malformations, and in parallel to understand brain development and function, which has been a very passionate interest of mine through my graduate work and beyond. And so in my lab, we study the natural history of different human conditions. So that's how patients present and how they change over time. Using the cohorts of patients that we enroll, we have identified genetic causes for a number of hindbrain malformation conditions. And then those genetic causes are a foot in the door for understanding the mechanisms underlying different human conditions. And we've done some work in that area. And then area where we haven't really gotten to is ideally identifying or developing treatments that will help the patients who participate in our work. And so why do we study these conditions? This is one of the main reasons. So these are all participants in my study whose parents have given permission to share their pictures. But I'm a pediatrician and helping kids is in my core. And so we love to get pictures. We send birthday cards every year to all our participants, now about 2,000. And they're really appreciated, and it keeps our families engaged with us and us engaged with them. So more tangibly, why do we study these malformations? First, we want to provide direct benefits. And some of those benefits, even without treatments, is that we can give more accurate outcome information and recurrence risk for families. We can monitor for complications that may not be apparent at the time of diagnosis and treat them earlier to give better outcomes. There are more and more disorder-specific treatments, whether it's targeting a genetic cause or a medication that works better or worse in a particular condition. And then other things that we don't always think about are some parents feel terribly guilty that they had a child and that with a developmental condition and that it might be their fault. And for many people, understanding that it's just that it's really complicated to make a human being and it's really complicated to copy DNA trillions of times, it can relieve some of their guilt and it provides connections with other families who have family members with similar conditions. So again, in parallel, I am a developmental geneticist by training, and so I would like to be able to contribute to understanding both rare and common human brain conditions. And some of our work has served as a model system for basic developmental processes, and I'll talk about that a little more. But first, what's the anatomy? And so I mentioned the tentorium, and the tentorium is a thick fibrous membrane that separates the hindbrain, particularly the cerebellum, and the upper part of the brain. And so most people talk about supratentorial things, so things that happen in the supratentorium, and there's less attention on this part of the brain, but in some ways, the hindbrain includes the brainstem, which mediates a lot of the most basic functions of the brain, including whether you stay awake or go to sleep. Allows you to change your breathing rate in response to environmental stimuli, regulates your heart rate, and then controls all of the muscles and sensation for the craniofacial area. The cerebellum historically has been thought of as an organ of motor control, and so it's very important for coordination and balance and things like eye movement and speech. But more recent work has highlighted its role in cognition and emotional regulation. And one of the unifying features of the cerebellum is that it may um, provide a signal processing or signal smoothing function across many different neurological processes. Um, and so let me make this smaller. So now I'm going to talk about some of the different categories of brain malformations to give you a sense of how we do this. And really, as a trained as a Drosophila geneticist, I viewed this approach as a human genetic screen where we don't control the matings, but we do identify phenotypes that we want to understand the genetics of. And so cerebellar hypoplasias just mean that this part of the brain called the cerebellum is smaller than it should be. And it can affect the midline cerebellum, which is what we mainly see here. But there's also two side regions of the cerebellum called the hemispheres that can be impacted as well. And it's interesting that some conditions affect only the vermis and others affect the whole cerebellum. Imaging category that's gotten a lot of attention and is a focus of Kathy Millen's lab over at SCRI is a vermis hypoplasia where the vermis is flipped up in the posterior fossa and there's lots of fluid below it. And it can be associated with other brain malformations, and that's again a very heterogeneous category, but that is one of the more common clinical presentations. And then finally, I'll talk to you a lot about a condition called Joubert syndrome that it has been the focus of my lab for most of my career. And part of that was we had a lot of success identifying genes that are associated with this condition. And so this is I showed you a Dandy Walker malformation image before. This is what Joubert syndrome looks like. There is extra fluid below the cerebellar vermis, but the cerebellar vermis is too small and it's dysplastic. And the roof of the fourth ventricle here is elevated. And I'll show you some additional pictures of that when we jump into Joubert. Another common category that's actually typically associated with quite substantial disability is what are called pontocerebellar hypoplasias, where the pons, so this is again the unaffected individual, this nice fat pons is nearly flat in these conditions, and then the cerebellum is much smaller than it should be. And so this is a particular category called pontine tegmental cap dysplasia that's exceedingly rare. Currently, we have about 50 patients enrolled from all over the world, compared to almost 1,000 for Joubert syndrome. And unfortunately, we don't know any genetic causes of this condition, so we really have no clues to the biology that underlies it. And then another focus of my lab has been rhombencephalosynapsis. And this is a condition where the cerebellar vermis is missing and the two cerebellar hemispheres are fused in the midline. And so you'll notice this sort of looks like a three-leaf clover here. That's what a vermis looks like. And here there really aren't those demarcations making it into a three-leaf clover. It really just looks like cerebellar hemisphere all the way across the brain. And unfortunately, that condition has also been relatively resistant to identifying genetic causes. And it's much more common than pontine tegmental cap dysplasia. We have, I think, almost 200 participants in our studies. So, a few, let's see, and then finally, cerebellar dysplasias. And so if you notice here on this axial MRI view, the cerebellum is folded and the folds kind of make this U-shaped pattern. And the cerebellar vermis is right here that I've been talking about. The cerebellar hemispheres are out to the sides. And cerebellar dysplasias have an abnormal folding pattern. And so you can hopefully tell that this individual who has Paredes-Boltzhauser syndrome uh, looks very different than this individual. And remarkably, those conditions have been much easier to identify genetic causes with diverse biology. And I'm not going to really talk about those today. All right. Any questions about the human conditions at all? I haven't really talked about the clinical features. Most patients present with what's called low tone, where the resting muscle tension is low, motor delays, where they're either have mostly discoordinated movements. Eventually, patients are often labeled with ataxia, and then they have abnormal eye movements, often trouble with making speech sounds and chewing and swallowing. Um, and those are the hallmarks of cerebellar conditions. Happy to pause for questions, but also we'll move on unless somebody entered something in the chat. Currently, I can't. Okay, I'm going to move on. All right, so we have been able to identify genes involved in different developmental processes, and this is sort of a mapping of diagrams of mouse development in a review that Kathy Millen and I wrote years ago, to genes that are involved in different aspects of cerebellar development. And the details aren't so important, except to make 2 points. One, unfortunately, this kind of human screen identifies kind of the least important genes involved in development, because the most important genes, if you knock them out or cause them not to function well, you don't get a viable even fetus, let alone human, child, or adult. And so I often say that, human genetics identifies the least important genes for development. But we do highlight important processes. So pattern formation in the early brain, um, proliferation and survival, morphogenesis, migration, cell fate, sort of in more the middle of brain development, and Joubert genes definitely are important for both migration and axon guidance. And then there are obviously genes involved in synapse formation. So this was very gratifying to be able to contribute to how human disease genes map onto human cerebellar development. All right. So more on Joubert syndrome. Like I said, this has been the big focus of my lab. And back to the imaging to give you a more detailed idea of how radiologists and people like me look at these images. The best way to diagnose Joubert syndrome is to look at what's called a sagittal or a side view. And as you can see, there's this three-leaf clover cerebellar vermis here. And on the lower image, I think I even drew lines. The vermis is only this part of the cerebellum, and this is actually cerebellar hemisphere pushing in from the side because there's no vermis to keep it out to the sides. And then one real hallmark of Joubert syndrome is this elevated roof of the fourth ventricle, which actually corresponds to something well, I guess I'll just show you, is these more horizontally oriented, thick superior cerebellar peduncles. And so those are the connections between deep cerebellar nuclei with the midbrain that really is the pathognomic feature of Joubert syndrome. So this is what radiologists look for when they're trying to determine whether Joubert syndrome, a patient has Joubert syndrome. And this is what normal superior cerebellar peduncles look like. They're much smaller, thinner, not as long. And I hope it's obvious, especially on the other images I've shown, that the molar tooth results from this top cutting surface here, which is mildly abnormal in shape, and then these roots of the tooth that are the SCPs. And then one other feature that's been very important is a superior cerebellar dysplasia. And so it's present in almost all patients, even patients with very mild forms of Joubert syndrome. And work, I think I even saw Hank in the audience, work by Hank Cheng in my lab, who's now a MD PhD student in Andrew Sturgakis's lab, uh, sequenced a bunch of patients without the molar tooth sign, but with features like this superior cerebellar dysplasia, and found that many of them, most of them, have variants in the same Joubert genes as cause this more significant and obvious phenotype. And that's a theme in genetic conditions, that there's a spectrum of any genetic condition. All right. So the other really interesting thing about Joubert syndrome is that in addition to the brain features, there's also features in subsets of patients across multiple organ systems. And so this is a picture of the retina. And so there's progressive retinal dystrophy in about 30% of patients. There's progressive fibrocystic kidney disease and or polycystic kidney disease in about 30%. There's a form of congenital liver fibrosis in about 12%, and about 14% of patients have extra fingers or toes. And so this was kind of a little bit of a mystery, although by the time we started identifying genes for Joubert syndrome, we had more understanding of pleiotropy. And when we get into the biology, it'll become pretty obvious why there's impacts in so many body systems. And so over the years, we've collected almost 800 families or enrolled almost 800 families, almost 1,000 patients now. And we've also collected patient fibroblasts. And I mention that because that's really intended to be a resource for the scientific community to do functional work in actual patient cells where we know that the patient had the condition and where the cell lines are linked to both genetic and clinical information about the patients. And they can be transformed into iPSCs and can be used for other experiments. And so one of the advantage over engineered iPS cells or other stem cells is that they have the genetic background that's present in the patient with the condition. And the map just shows that our patients are from all over the world. And so this is a history of gene discovery in Joubert syndrome. And again, the details aren't important. We're now up to 43 genes. We're tailing off. Exomes really helped back in the late 2000 aughts. And with current exome or genome, we can explain about 80 to 85% of Joubert syndrome. And in the remaining patients, Some have single variants in known Joubert genes, and others have no variants in known Joubert genes, and that's important because it tells us where to look for the remaining heritability for Joubert. And the kinds of things that we're continuing to look for are new Joubert syndrome associated genes. And so there's a new gene that should be hopefully coming out this year, but maybe next year, the last one in that graph. Difficult to identify variants, so either because they're non-coding or repeat expansions or mobile elements and structural variants, and most of these have been identified in rare patients with Joubert syndrome, so we're finding those. We also have discovered that there are some examples of non-recessive inheritance where only one variant or a variant in only one copy of the gene is required to cause Joubert syndrome. The only example of that is the SUFU gene. And then oligogenic inheritance, where variants in more than one gene can cause Joubert syndrome, is hypothesized, but there really aren't any firm examples yet. And I absolutely think that it happens, but it really muddies the water right now because we don't know when it happens and with which genes. And so genetic counseling often has to involve the possibility of oligogenic inheritance, which is much more complicated than simple recessive inheritance. So we don't know. All right. So where did that leave us as we sort of completed understanding the genetics of Joubert syndrome? It really meant that we understood a lot about DNA defects, and as complex as the genome is, it's not as complex as an organism. And so this was sort of on the simple end of things, although it seemed complicated at the time. And that's told us a fair amount about what kinds of protein defects and protein dysfunction cause Joubert syndrome. So we understand some, but less than the DNA at the DNA level. As far as cellular defects, I mentioned that, well, actually, I haven't mentioned yet. So this is an interesting point that I'll get into where each time a Joubert gene has been discovered, in general, new biology or different biology than prior genes has been published along with it. And I think this is a pretty clear example of publication bias because I think that the biology relevant to Joubert syndrome should be somehow unified across genes. And so really people should be publishing the new gene and saying, look, it does these similar things to the prior genes, but that doesn't get you into as high profile a journal. And so what the rest of my talk will talk about is our work looking at the different cellular or some cellular defects to try and sort through what's going on. And then really, while I trained as a developmental biologist, I did not continue animal model or work in my lab, and so we haven't done any of that. And so the central question that's tormented me and people in my lab for quite some time is how does dysfunction of so many different genes lead to the same specific Joubert syndrome brain phenotype? And the corollary to that is, do all the genetic causes converge on a single cellular mechanism? And so we've touched on the fact that Joubert syndrome is a ciliopathy, and that hopefully all of you have heard of primary cilia and that they're really important. But when I started this work, we barely knew what they were. There was some work in the 1960s and on, sort of looking at them as potential vestigial organelles that used to be evolutionarily motile cilia, and they are certainly related to motile cilia, but they have many distinct functions. And so looking at the protein localization of different Joubert genes really pointed directly to the cilia. And so this is one gene that we identified not so long ago that encodes a protein that's at the basal body of the cilium. So I'll show you what the different major regions of the cilium are in more detail in a subsequent slide. But that and many other genes pointed directly to the cilium. And actually, I'll tell you the parts of the cilium now. And so the cilium has a basal body that anchors it to the cell. It has a transition zone that basically regulates the protein composition of the cilium, allowing some proteins in or facilitating their entry, and doing the same with ciliary protein exit. And then there's a long microtubule-based axoneme that has a variety of proteins associated with the microtubules, and there's a lot known about the transport processes that gets proteins in and out of the cilium. And then the cilium tip is actually a remarkably complex region that's involved in a number of signaling pathways. And the ciliary membrane is decorated with a variety of receptors for signaling pathways. And so this is a map of the different Joubert associated proteins with the different regions of the cilium. And so essentially all of them are in and around the cilium. So that was a great clue. And I was really happy that I didn't end up studying human biochemical, like a Krebs cycle disorder, because that would have been really uninteresting to a developmental biologist. Cilia are much more interesting. Little bit more about cilia. So they are remarkable organelles. They appear to be involved in mechanosensation. So if you bend them, they change their signaling. And that happens in the kidney and actually during bone development and probably many other places. They're also how we smell. So they're chemosensors, and they are on the olfactory neurons in our sinuses or a cribriform plate. And they have olfactory, they're studded with olfactory receptors. We also sense light through highly modified cilia. So our photoreceptors are modified cilia packed with opsins that sense light. And then, as I've alluded to, primary cilia are very important for developmental signaling and allowing cells to sense what's going on outside of them and respond appropriately. And then this is just a diagram to highlight the differences uh, that between motile cilia that have central to a central doublet of microtubules and motor proteins that allow them to move. And most primary cilia do not have the central two microtubules and are not motile. Although, just like everything in nature, there are exceptions to the rules. And so when you screw up cilia, what happens? Well, you can have a variety of different conditions, and it depends on which proteins you are defective. And so these conditions, many of them are largely overlapping, non-overlapping, although it's turned out that Joubert syndrome and Meckel syndrome are just the mild and severe ends of the range of dysfunction for the genes that I've shown you on previous slides. And some of the genes are also cause nephronophthisis and even isolated nephronophthisis. So there's some overlap with this kidney phenotype. That's the fibrocystic kidney disease. And then there are a variety of other conditions that are really genetically non-overlapping with Joubert. More important than knowing the names of all the conditions is sort of understanding which body systems can be impacted. And really, Joubert covers a lot of them. So the brain, the retina, the kidney, the liver. There can also be skeletal dysplasias where the bones don't grow correctly and are often quite short. We talked about extra fingers and toes. Laterality defects can be seen in severe ciliopathies because cilia are essential for left-right asymmetry formation in humans and many vertebrates. And there is some overlap with motile cilium defects. So there's a whole nother class of conditions called primary ciliary dyskinesias, where the cilia in our lungs and other places aren't motile or aren't as motile, and that means that patients can't move mucus out of their lungs or their noses or their ears, and they get infections in those organs, and it can be a very difficult to treat condition. And then the role in obesity, there's been a fair amount published about how cilia regulate weight. I don't know that we know the final answer on that, but there certainly are some conditions, ciliopathies that are associated with obesity. All right. The combined prevalence, they're pretty common, largely because of autosomal dominant kidney disease, which is the leading genetic cause of kidney failure in adults. So before I move on to more mechanistic work, I thought I'd pause again to see if anybody has a question. Happy to field it. It's always good to know you're still there. But hearing none, I'll forge on. And so I've mentioned that I think that there's some publication bias for highlighting novel aspects of biology associated with dysfunction of each Joubert gene. And what has been published is defects in ciliary number, so how often cells make cilia compared to normal. Cilium morphology, either cilia can be too short or too long or curved or have other defects. People have looked at cilium protein content, and a number of the Joubert genes are involved directly in or required for the transport of other Joubert proteins into cilia, almost in a kind of pathway uh, um, configuration. And then people have also noted that the post-translational modifications on ciliary microtubules can be altered in Joubert with loss of Joubert gene function. And then there's been a lot of work on different signaling pathways, primarily hedgehog signaling, which has been shown to be abnormal in a variety of conditions where the Joubert genes are not functioning correctly. There's been some links to DNA damage response. I don't really know what to do with those studies, but at first I thought it was a red herring, but there's been a number of additional publications where there seems to be some consistency to it with a subset of Joubert genes. So it's an interesting and hard for me to think about area of biology. And so this was very frustrating to me, as I mentioned before, and to my lab. And so our central question is, how do all these genes, when they're not working, somehow impact the cilia through what we hypothesize is a central mechanism, leading to this very specific human brain phenotype. And I think the reason, what's convincing to me about why it's so specific is that we really can tell what genes are going to have variants in them when we look at the MRI. And it's been consistent in an essentially blinded way because we enroll the patient and then do an exome and look everywhere. And we've essentially always found cilium related genes. I think there's going to be a lot of more complex genetics and biology about how all the other phenotypes are formed. And we would have gotten different sets of genes if we anchored on different phenotypes. So people who look at retinal dystrophy find the Joubert genes, but they also find other genes that don't cause Joubert in patients who don't have Joubert. Similar with defined forms of these other phenotypes. So why go for the Holy Grail? Because the best treatment targets will be shared across genetic causals. All right, enough of the Holy Grail. So, what are our primary candidates for this hypothesized central mechanism? Well, one of the genes is called INPP5E. It's a phosphatidylinositol phosphatase. And there have been experiments showing that it actually changes the PI composition of cilia. And so that when you have an enzyme involved in your condition, it's hard to ignore that enzymatic function might be important. And so that's one thing that we've looked at some across genetic causes. A lot of the Joubert proteins localized to the transition zone, most of them, in fact, half of them. And so we think that ciliary protein content may be the core mechanism of Joubert, and so that's an area of interest. And then hedgehog signaling has been shown to be aberrant across many different models for dysfunction of the Joubert genes, and so we can't ignore that one either. But I, being trained as a geneticist, really value taking a less biased look by doing screens rather than trying to use my limited cognitive function to identify the right path to follow. And so I was lucky enough to have a cell biologist join my lab, Julie Vandewey, who came for a postdoc. And kind of remarkably and a little alarmingly, she agreed to do a big proteomic screen in a library of cells knocked out for 13 different Joubert genes. And so this was kind of a heroic endeavor that spanned the pandemic and is ongoing because we still don't, we're still picking through the results. And so the experimental design was to isolate cilia from engineered cells with knockouts for a library of Joubert genes. And to do that, she adapted a protocol from mouse cultured cells. And this is before, and she decided to use human HEK cells because she tried probably 10 different cell lines. And these were the ones that deciliated the best. They have lots of issues, but it was essential to be able to get a good source of cilia. And so this is pre-deciliation. This is RL13B, actually one of the Joubert proteins that marks cilia really well. And after deciliation, the cells are still there. There's a couple cilia, but most of them are gone. She did a lot of work looking at the time course for deciliation, so it's quite fast, which is good because we want to keep them intact and not have things leak out as much as we can. And then she did a number of known markers of different parts of the cilium and looked in the whole cells before deciliation, the cell bodies after deciliation, and isolated cilia. And so what you can see here is that the cilium prep is highly enriched for cilia. I would not ever say it's pure cilia. The cell bodies still have some residual cilia on them, and both the whole cells and after deciliation. And that's because we don't get complete deciliation, but it works pretty well. And when you take a picture of the supernatant, you can actually see linear green structures. And so we wanted to quantitatively compare ciliary proteins and I'm just checking the time. Okay. In mutants versus controls and look for proteins that were altered across multiple different genetic causes of Joubert syndrome, hoping that that would lead us to a shared mechanism. And so this is just the experimental design with replicates for all of the different genes up here. And she did 13 genes. 2 of them we had technical issues with, but the rest we were able to CRISPR, usually biallelic frameshift variants into the hex cell lines. And luckily, because hex are largely triploid, for the ones that were triploid, we were able to get all 3 copies. And then we partnered with Mike McCoss, who is an amazing proteomics expert in genome sciences at UW, who has a technique called data independent acquisition for measuring proteins in these kinds of samples. And I will not pretend to be in any way a proteomics expert, but obviously his running the samples and helping with the analysis has been essential for this work. So what did we find? We detected almost over 5,000 proteins in these preps, And when we used an adjusted p-value of 0.05, we had many proteins that were both more highly localized or less localized to in our cilium in the mutants. And the distribution of how highly they were enriched or decreased in the cilium is that's what these graphs show. So proteins that were increased are on this side, and proteins that were decreased are on this side. And so then we took those proteins and looked at how many cell lines had that protein altered in them. And so we had 1,200 or so proteins that were only mislocalized in about in one cell line, and about 900 in two. And really, we were most interested in the ones that were over here, but we didn't find any, which is not surprising. And Julie is now focusing on the top 30 proteins that were shared across the most of our mutants. And this is the list. Most of them, interestingly, were accumulated, which I don't know that we have a hard theory for why that is true. It could be a technical issue with it being easier to identify things that are overrepresented. A few are underrepresented or under localized. And it's been heartening in a few ways. One, we've taken 30 proteins, random proteins from the entire proteome, and they don't look anything like these as far as their relationship to cilia. So we're very confident that we're pulling out cilium-related proteins. And the functions of these proteins fit with some of the models for Joubert syndrome. And so this is a particularly interesting protein called oxysterol binding protein like 2, and it's localized to the base of cilia, and it's involved in exchanging phosphatidylainositols for sterols in protein membranes. And that's quite tantalizing because of the involvement of the INPP5E gene in Joubert syndrome. So direct link to phosphatidylainositols. And then hedgehog signaling. Cholesterol is very important in hedgehog signaling, both for the ligand and the reception of signaling. And so we're very excited about that protein. We have a mystery set of proteins. So the prefoldin complex is involved in tubulin processing, but has never been shown to be in cilia to our knowledge. And our cilium preps do include the base of cilia, so prefoldins might accompany tubulins to the base of cilia, and they're important for allowing tubulins to fold and polymerize, among other functions. But this is really a novel association that we're also or Julie's lab is very interested in pursuing, because all the work is going to be done there. And then definitely a number of proteins that are involved in hedgehog signaling. And so those are being pursued. And I mentioned that tubulin post-translational modifications are altered in Joubert mutants, and largely thought to be through the involvement of HDAC6, which is a deacetylase. But the fact that we found 2 out of 30 acetyltransferases that were not known to live in cilia was interesting. And so Julie's lab now is looking at the localization of these proteins, and the ones where there are good working antibodies. She's had good success localizing them to cilia. For the others, she's making tagged versions to look at their localization, both in controls and mutants. And so I have to say that I was really worried for several years while she was doing this work that we would come up completely empty, but I think we have some very attractive things to pursue. And even if they're not the answer to Joubert, they're revealing new biology about the psyllium. So, all right. So in summary, for at least the second half of the talk, I hope you take away that Joubert syndrome is a very specific ciliopathy phenotype associated with a very specific set of genes, but it's very heterogeneous, that there are a number of proposed mechanisms, um, and that we hypothesize that there is a single underlying mechanism, although we're doing the work to be convinced if it really is that we're getting a convergent phenotype with fundamentally different underlying mechanisms. I'd like to thank folks who've contributed to this work, my lab here at UW and Julie's now at the University of Oklahoma. She left, I can't remember, a couple years ago. And we have got funding from a lot of different sources, both grants to our labs, but also the now Human Institute on Human Developmental Disability Genetics Core helped us quite a bit, and the University of Washington Center for Mendelian Genomics, which is now the Center for Rare Disease Research. All right. Thank you very much for your attention. I'm happy to answer any questions.
Thank you so much, Dan. Very beautiful work. Let me see if there is any questions on the chat, and I will be checking. If anyone wants to ask questions, please unmute yourself, and I'm going to start. I have a few, but let's start with the end of your work. So for the protein, for the top 30 proteins that you guys discovered, so did any of those are actually mutated in causing disease already known, or they can be like causing disease, like a variable phenotype and things like that contributing to this in Joubert, do you think?
Right, so none of them are known Joubert genes.
Mhm.
One of the things that we're interested in for the proteins that are upregulated, you might imagine that they might be dominant causes of Joubert syndrome and only require a variance in one gene. So genes that gain a function variants tend to be only in one copy of a gene or only needed in one copy of the gene. And so we did look at our cohort in the unsolved families, which aren't very many, to see if there were variants in any of those genes in our cohort, and we didn't find any. I haven't systematically gone through to see each gene to see if it's associated with any human disease, but the proteins have not been associated with other ciliopathies, which is interesting
Mhm.
directly. Like variants in those genes haven't caused known ciliopathies. Um, so somewhat surprising, especially because when you lose RL13B function, INPP5E doesn't localize to cilia as well as it normally does. And so we didn't see INPP5E very well in our proteomics data. The peptides just weren't detected. And so, but you might have expected for other proteins that are involved in known ciliopathies that we would have seen them.
So you mentioned like gain of function. I was wondering, I don't think you mentioned that, maybe I missed during the talk, but what is the main mechanism that cause diseases? I say is recessive, but it's like alteration in the protein function, or is like complete loss of function?
It's a mix. So I have a slide sort of showing each gene. So INPP5E almost never has truncating variants in it. It's almost always missenses versus a gene called CP290 where it's almost always truncating variants. And so different genes are differentially vulnerable to different types of variants. But we think they're all loss of function. We think the one dominant cause is actually a haploinsufficiency mechanism, not a gain of function.
Okay.
Looks like
Amy?
Amy.
Yeah, hi. That was a great talk. Thank you so much. I was interested in the tubulinopathies. When you have something that's so highly conserved, like a tubulin, is there some pattern in terms of where variants associated with specific disease are associated on the protein structure, or, is it levels of protein, or is it just all over the place?
So the answer is it's often very specific. And I probably know the neurologic tubulinopathies the best because we've identified the conditions associated with some of those genes. But it's not a simple clustering, like linear clustering in a domain, just so you can see it when you have a linear protein diagram. A good example of that actually is not a tubulinopathy, but we identified in a gigantic protein called MACF1 variants in one particular binding domain, but they're separated by hundreds of amino acids, but they all come together to make a binding pocket. And there are four residues that are mutated for this one very specific phenotype. And um, it we had an interesting experience where a collaborator said, "Oh, well, we have a deletion in the gene." And we said, "Well, we predict your deletion is going to take out just one of those four residues." And it turned out that that was the case. And so, without knowing what the variant actually was. And so there are very domain specific conditions where you have to hit very small targets. Another example is a skeletal dysplasia called achondroplasia, which is largely caused by a single variant, but it's a very common skeletal dysplasia. So that's a form of, I think, the appropriate term is the most common cause of little people, and that's the name of their national organization. So, um, but it does sometimes point to very specific biology. Joubert syndrome tends to be just generic loss of function for the whole protein.
Thanks.
There is a Michelle, do you want to unmute yourself? There is a question in this chat.
Um, yeah, okay. Thank you for sharing your work. It's very interesting. I am not a developmental biologist in any means, so I'm actually curious about the thing that you said that well, okay, I'll get to my question. So the so your proteomics data showing that like the top hits tend to be overaccumulation or overaccumulation of proteins, which I find surprising. Um, and although you're looking for genes relating to cilium development, could the data also point to something relating to metabolism, like misregulation of some sort of like lipid or yeah, lipid
Mhm.
metabolism or maybe like autophagy?
So the short answer is yes, although I am not an expert on what you would expect if you saw sort of a general autophagy kind of dysfunction. But cilia, there have been a fair number of papers linking cilia to autophagy. And I have to admit that I don't know the details of that literature, but we are mystified by why we got so many that are accumulated rather than depleted. And I think that is a good question as part of figuring it out. Um,
Thank you.
Okay, any more questions?
Hey Dan, it's Hank here.
hey, Hank.
Hey Dan, I was wondering, for the around 20 to 30% of patients that are still unsolved, I I know that it could be due to a lot of reasons, like maybe there's some non-coding variants, maybe there's structural variants I just can't detect with exome sequencing, or maybe there's like oligogenicity. And I was wondering, do you have any hypotheses on which of these mechanisms might be explaining most of these unsolved cases?
Yeah, I wish it were oligogenicity because I think it would be really interesting, but I mean we're down to 20-ish% where we're not finding much for candidates for oligogenicity. So I think that's going to be exceptional, but I think it's all of it is going to be exceptional. And I think we got really spoiled when exomes came around and we were able to suddenly solve like 30% of the well-defined conditions. And a high percentage of the ones where we knew which genes to look in. And now we're slogging through, as you well know, the much harder to find variants. And I think it's going to kind of be all of the above. We're going to have a little bit of each, and that's what we're seeing so far. And I'd be interested, obviously, you and Andrew's lab are doing a lot of that. Does that reflect your experience across other conditions?
Yeah, I think that's a great question. I mean, we haven't solved that many. The ones that we have solved are non-coding variants that were missed by exome. Yeah.
Mm-hmm.
So if I were to have a hypothesis, I think if we were to do like genome-wide sequencing on your Joubert syndrome patients, you could find some like cryptic splicing variants.
Yeah, we did it on about 90 so far, and we found a bunch of CNVs that, again, it was the exomes were done a while ago, so CNV calling wasn't, copy number variant calling wasn't as good for finding small deletions. And we found a bunch of non-coding variants, but it's hard to sort through them without having cell lines or putting them into cell lines or doing splice assays or things like that. So that's definitely borne out. It's just not like of the 20% that we have left, 3/4 of them don't have obvious splice variants, which would be good because then we would be very nearly completely done, instead of sort of limping along, finding a variant here or there. So,
Yeah.
um,
Yeah. Thanks, Dan. Yeah. That makes a lot of sense. Thanks.
but I'm happy to provide what we have if you and Andrew want to chew on them.
Yeah. Thanks. I think if I don't know, that might extend my PhD by too long. So, back to you on that. Yeah.
No doubt.
I actually have a question for both of you. So for the no-coding variants that I think is Hank, right? You guys are finding, are they causing gain of function or loss of function? Like lower expression or higher expression?
So the ones that we're finding, they are loss of expression. That's just been my experience. Although I think certainly you could find gain of expression cases. I think it's just like in the literature right now, a lot of the disease-causing variants are loss of function because I think they're just a little bit easier to detect. Like if you do RNA sequencing, you find loss of expression, you find that as an outlier, and you can say like that's the mechanism. So a lot of the literature out there focuses on loss of function genes, and so that kind of biases you to when you're looking for new genetic causes. They're also going to be loss of function. They might be a non-coding variant, but they're going to have the same mechanism. So far, we're still detecting loss of function. Um, yeah.
Okay.
Yeah, and I
Thank you.
don't know that I know of any non-coding variants that are gain of function. I'm sure there are examples because there's always examples of everything you can think of, but it's certainly not something compared to loss of function. It's not a common occurrence that we know of, with the caveat of I'm not sure we have looked as carefully as for loss of function.
So we have a patient that we are working right now where an inversion disrupted a region kind of 80 kb apart from SOX3 and caused sex reversal. And the gene is actually high, it's gain of function is kind of overexpressing the gene, so it's kind of interesting.
Yeah.
So
Yep.
yeah, okay.
Well, and I should say, well, there are almost certainly gene fusions that do similar things as well, in comparison to regulation. So, yeah, I'm sure I'm ignorant of some of the examples of gain of function caused by non-coding variants.
Okay, any other questions? Otherwise, oh, thank you again, then.
Sure, my pleasure.
A great talk. We'll talk later as well. I wanted to just comment on the next Science Matters seminar series, which will be November 19th. And we will feature Dr. Isidro Cortesiriano from the group leader of the European Bioinformatics Institute, that he will talk about cancer genomics. Thank you, everyone.
All right, thanks. Bye.
Bye.